Okay, guys, so today we are starting Chapter 4, Sensation and Perception. Um, Remember that this is a video, so I'll probably talk a little bit more quickly than I do in class since I don't have you in front of me to cue me in when I'm going too fast. So just remember you can pause to catch up with your notes. You can always rewind and rewatch slides that maybe are confusing. So now that we understand a little bit about how the brain works, we also need to understand um, how the brain is getting all the information that it responds to. So that's the study of sensation and perception. There's a big philosophy of science question that revolves around this area of study, and that question is, if you just had a brain in a jar, just a brain alone, is that enough to create the human mind? And a lot of modern philosophy of scientists theorize that the mind is created by the brain's response to and interpretation of the environment around it. So without the input of sensation, there is no human mind that gets created. So let's talk about how this happens. How does all this information get to the brain, and what does the brain do with it once it arrives? Some other really big questions in the field of sensation perception are what we call the binding problem. And that is basically the problem that we don't understand yet how the brain completely recreates our experience of the world. We'll go through how different parts of our sensory experience are processed. We've already talked about the concept of association areas where different parts of information get recombined. But ultimately, we don't have a spot in the brain where we see everything getting recombined to create our entire perceptual experience. So this is a question that is unanswered in the science of psychology right now. We also are constantly investigating how much of our experience, our internal perception, actually reflects reality. So I teach a whole semester on sensation and perception, and that's an area we really focus on, is highlighting when our brain is creating our perception and that perception does not reflect how the stimuli actually exist in reality. So we'll have some examples of that as well. All right, so before we talk about any specific sense system, I want to talk about some basic principles that are true of just studying our senses. So if we refer to sensation, that's just a simple awareness at a receptor in the body due to the stimulation of a sense organ. Right, so we'll start with vision. So sensation for vision is when we see cells in our retina responding to light waves. Perception is the organization, identification, and interpretation of a sensation in order to form a mental representation. So basically, when sensation happens, there's this responding of a receptor organ, and then our brain interprets that signal that it gets from the receptive organ and forms some kind of mental representation of the stimulus that it's responding to. The process of communication between these two steps is called transduction. So transduction is when the sensors of the body convert physical signals into neural signals. Right? So it's the process of the retina turning light waves into action potentials that the brain can interpret. Okay, so without transduction, there can't be perception. And then as we look across our different senses, we can study what's called sensory thresholds. So we have what's called the absolute threshold, which is the minimal intensity of a stimulus needed to detect it with 50% accuracy. So if you could imagine if you were in a pitch black room and I was showing you little lights and I was making them dimmer and dimmer, your absolute threshold would be the minimum amount of light I could show you, and you'd still be accurate 50% of the time to be able to say, I see that light, and you'd be correct when you say that. Now, you would expect that human thresholds would look like the graph on the bottom left, where there was a set intensity where we would always detect it, and then a set intensity where we would never detect it. But what we find is that humans respond like the graph on the right. That S-shaped curve occurs again and again when we test people's sensitivity to stimuli. And what you can see is there is basically a middle range where people are likely to miss stimuli that are actually there, and also sometimes report detecting stimuli that actually aren't in the environment. So some examples of, on average, sensory thresholds for humans. For vision, we can see a candle flame 30 miles away on a clear, dark night. Right? So that's a pretty sensitive process. For hearing, we can hear clocks tick 20 feet away when it's all quiet. For touch, we could detect a fly's wing touching our cheek from 1 centimeter away. For smell, a single drop of perfume diffused through an area equivalent to the volume of 6 rooms. And for taste, a teaspoon of sugar dissolved in 2 gallons of water, we would still drink that water and say, "This tastes sweet to me." And so just some examples of the bare minimum for different types of stimuli where we'll still be able to detect that it's there, again, 50% of the time. We also can test for difference thresholds. So this is when you can detect a change in a signal. So you already know a stimulus is there. How much do you have to change the stimulus for you to consciously perceive that it has changed? That minimal change needed is called the just noticeable difference. And it is, there is a formula for figuring out the just noticeable difference called Weber's law, which is just that the amount of change you need in a stimulus to detect that change is proportionate to the original intensity of the stimulus. Okay, so for example, if somebody is speaking at a whisper, they don't have to increase the volume of their voice very much for you to notice that the loudness has changed. Right, so if you start with a weak signal, a whisper, you need a small change in order to notice it. But if somebody starts yelling at the top of their lungs, they have to pretty much double the volume of their voice in order for you to notice that it got louder. Right? So if they start at this super loud volume, you need a really big change in order for you to notice the change. Okay, so the amount of change needed to detect the difference is proportionate to the original starting value of your stimulus. We also can look at signal detection theory to predict when people will be aware of a stimulus in their environment versus not. So we have 2 principles here. We have individual differences in sensitivity to different stimuli. So some of us are more sensitive to light than others, some of us are more sensitive to sound than others. So our thresholds are going to be different across people. But then we also have individual differences in response biases. Okay, so if we look at the bottom left, we have the possible outcomes if you're doing signal detection theory, which is just the likelihood that someone will detect a stimulus if it's present. So on the top we have a light is presented and the participant says, "Yes, I see the light." We call that a hit. If a light is presented and the participant says, "No, I don't see a light," we would call that a miss. If we don't present a light, but the participant still says, "Yes, I see a light," we call that a false alarm. And then if the light is not presented and the participant says, "No, I don't see a light," we call that a correct rejection. So those are the 4 possible outcomes when you're just asking people if they detect stimuli or not. And what we found is basically there's this psychological difference in people called response bias, where people basically range from liberal to conservative in the likelihood that they'll say yes to a stimulus. So some people just seem to be really comfortable saying, "Yes, I see something," and they don't mind being wrong. So in the case of a liberal criterion bias, their hit rates get really high because they almost never miss a stimulus when it's present, but their false alarms also get really high because they frequently say they see something when actually nothing is there. People who are higher in a conservative criterion response are really uncomfortable with being wrong. Right? So they're really conservative with when they're willing to say, yes, I detect something. So in that case, their misses get really high. So stimuli get presented and they say, no, I don't see them pretty frequently. But their correct rejections are also really high. So they almost never falsely say, yes, I see something there, when nothing is there itself. Right, so two different individual differences that are going to influence the likelihood that you detect a stimulus in the environment. And then, no matter who you are, our senses adapt, which just means that our sensitivity to a stimulus tends to decline over time. This is a good thing because it means that we're not constantly distracted by all the stimuli that are present throughout our days. So for example, when you got dressed this morning, you probably had some awareness of the softness of the clothes you put on your skin, their texture, their weight, but until I brought your attention to it just now, you probably haven't thought about that again. Right? So the receptors in your skin for that kind of information basically sensitize to your clothes and stop responding to them. So you don't get distracted by that information all day. Another example is if you're sitting in a chair, you probably stop being aware of what the chair felt like in your legs until you paid attention to it just now. So we also we sensitize to the taste of our own mouths. I know that's gross to think about, but imagine if we were consciously tasting what our mouths taste like all day. Also, the smells around us, we each have our own odor. We're usually not very aware of that odor, but you're probably aware of the odors of the people around you. Right? So we sensitize to our own smells, etc. All right, so if we look at the route for sensory processing in the brain, all of our senses follow this common model. So on the left, it shows that first there's some kind of receptor organ, your skin, your retinas, your inner ear, whatever it is that responds to a stimuli from the environment. They send information to the thalamus, right, our big sensory relay center, and the thalamus will send that information to the primary sensory cortex for that stimulus. So it could be primary visual, primary auditory, somatosensory cortex, wherever that information needs to go. From primary sensory cortex, information goes to secondary sensory cortex, which is just a more complicated level of processing for that stimulus. And then that information gets sent on to association cortex. So this is kind of a simple graphic for understanding the flow of information, but we are talking about the human brain, so of course it's much more complicated than that. So on the right is a better demonstration of the current model, where we've realized that each level there's multiple different nuclei for different types of information. We've realized that within each level, different nuclei can talk to each other, so there's a lot of parallel processing. And we've also realized that some pieces of information can skip levels, and so not all information goes through every single one of these levels. And really, I need to update this because now we've discovered what are called cortical fugal pathway, which means cortex down, where basically the highest levels of association cortex have arrows that go back down and can influence the sensitivity of even the receptors in the body. And so basically, as most things in psychology end up, it's the most complex version of this model possible that is accurate. Alright, so that's kind of a general framework for every one of our senses. And now we're going to go through the sense of vision in a lot of detail. And then next week when I'm back in class, we'll go through our other senses in not quite as dramatic detail. So vision. So vision is the most studied sense for humans, and that is because it has been recognized as so important for human experience that it's the most traumatic sense to lose. Right? So people who go deaf, who lose their sense of smell, who experience paralysis and can't feel parts of their body, they are psychologically better off than people who lose their sense of vision, that losing vision is a very traumatic experience. So because of that, we have studied it very aggressively, because the long-term goal of psychology is understanding human behavior so that we can optimize human behavior. So if we know that losing vision is really traumatic, if we can recreate vision for people, then we would be doing a lot of good for human psychology. So this is the cover of Wired magazine from September 2002, and it's showing some glasses that were developed at that time that have a photoreceptor in the lens that can detect light from the environment, and as you can see, it goes, there's a wire that runs from that photoreceptor and is actually wired into the person's occipital lobe. So this was some of our very early attempts to artificially create vision. And when we get to the end of the vision section, I'll show you where we're at today. So, in trying to understand how vision works, first, there is kind of this phenomenon where the input for vision is actually really crappy. Right? So everything that we see, if it is a 50-foot tall tree, or if it is a paperclip on our desk, it has to get distorted in size so that the light it's reflecting fits through our pupil, which is a couple millimeters big. Right, so all of our vision gets inputted as this weird little shrunken version of itself. And as soon as it enters the pupil, it hits this lens, which looks just like a contact lens behind the pupil, and it flips that the reflection of that object upside down and mirrors it, so that things on the left get put over on the right, and vice versa. And then that light hits the back of our retina, which is a little bit curved, but when we're talking about the light representing a single object, it's hitting a pretty flat spot on the retina. And so we have basically a 2D input of all of the 3D information we can see. So you can imagine if there was another candle behind the one in this slide, that its light would be smashed right on top of this other candle on the same spot on the retina. Right, so we get this distorted, shrunken, upside down and flipped 2D image that gets sent into the brain, and yet somehow the brain is able to recreate this dynamic and detailed 3D perception of our visual world. So we're going to try to understand how that happens a bit. So first, let's just talk about light. So light is necessary for vision. There are no species, no animals that can see in the dark. But there are differences in our sensitivity to light across animals. So you've probably noticed if you have a cat, that your cat can run around your house at night with little difficulty. That's because cats have a very high sensitivity to light, so they need very little light to be present in order to see. Now, if you truly put a cat in a pitch black room where there is absolutely no light, they will be just as blind as we are, right? So they have to have light to see, they just can see with less light than we can. Alright, so light can be considered in two different ways. You can think of it as photons of energy, or you can think of it as waves of electromagnetic radiation. Right, so each of those are useful for understanding how our retinas respond to light. And there is a spectrum of light that humans are able to detect. And that's light with wavelengths between 380 to 750 nanometers in frequency. And so let's look at this graph. So you see gamma rays on the left and AC circuits on the right. So this is all the type of electromagnetic radiation that's popping around our environment. If we were in the classroom right now, there's tons of X-rays and radio waves and AC waves from the electricity in our walls that are bouncing around the room that we don't have receptors for, so we are not conscious of all of that information. There is this little band of the electromagnetic spectrum that we do have receptors for. So this little band is blown up here, and this is what we call the visible light spectrum. Okay, so you can see it runs from that 380 to 750 nanometers of wavelength, and that's just referring to how fast the waves are coming in for this information. The slower ones we perceive as more purple and blue, and the faster ones we perceive as more orange and red. Those wavelengths of light have several pieces that we perceive as information about the light. So the length or the wavelength of the light, which is the distance between the peaks, we perceive as the color, right, or the hue. So again, if these were very slow and far apart, we would perceive it as more blue, and if they were really close together, we would perceive it as more red. The amplitude of the waveform, or the distance from the peak to the valley, we perceive as the brightness. So really high amplitude waveforms are very bright lights, and very low amplitude waveforms we perceive as dim lights. And then how symmetrical the pattern of the waveform we perceive as the saturation or the richness of the color. So if you have a perfect sine wave like we have here, we would perceive that as like a perfectly pure red. We almost never encounter pure colors in our natural environment. Usually it's some kind of mixture of waveforms, so you get changes in the frequency and the amplitude. So if you imagine if you're looking at a tree's trunk and you have kind of that mottled brown color, that's because you're not getting a pure waveform like this that represents brown. You're getting a combination of a bunch of different types of browns, different brightnesses, different hues, and so that's what makes it look like not a super saturated color. All right. So once this light gets in the eye, as I mentioned, it's going to go through this lens. And the job of the lens is to focus light on the retina. In order to keep light in focus as we shift our gaze from stuff that's close to us to stuff that's far away, the shape of the lens has to change. So there's these little ciliary muscles that are surrounding the lens that can flex it and make it more concave, so push on the sides, versus pull on the sides and make it flatter. And that process of the lens adjusting its size to keep images into focus on the retina is called accommodation. Okay? So just if you can imagine having like a zoom camera, if something is far away from you and you do that zoom in feature, that will literally move the lens forward so it's closer to the thing that you're trying to keep in focus. So if you're looking at something far away, these ciliary muscles will pull on the lens in your eye, which will bring the back of the lens closer to the pupil, right? So it's literally moving the lens closer to the thing you're looking at. If instead you switch your gaze to something close to your face, those ciliary muscles will push on the lens, making it more concave so that the back of it moves further away from your pupil, right? So again, that's just accommodation, that adjusting of the lens to bring things into focus. So the brain can actually monitor the activity from these ciliary muscles to infer depth. So it knows that if the lens is flat, you're looking at something further away, and if it's really concave, you're looking at something close. So that's one way that the brain can start to add back in the 3D information in our vision, is by adding in the input from these muscles. We also have two binocular cues that help us with depth perception. Binocular cues just mean that they're created by the fact that we have two eyes. So our eyes are set a couple centimeters apart, and they have to converge when we are looking at objects that are close, which basically means that as you shift your gaze from something far away to something close to your face, your eyes have to turn slightly inward, or they converge towards each other. And so the brain can monitor the muscles that move the eyes, and when they detect that they're converging, it knows that there's some depth involved, right? That something's getting closer to you than it used to be. We also can look at binocular disparity, which is the difference between the images on the two retinas, right? So when you're looking in the distance, the light that's shining onto your left retina is slightly offset from the light that's shining onto your right retina. The brain can compare the images on the two retinas, and if there's a lot of disparity, meaning that there's not much overlap between the images on the two retinas. It knows that that means that there's something far away. Versus if you're looking at something really close, the images on the retinas start to overlap more and more, or the disparity decreases, and that we interpret as something being closer to us. Right? So both convergence and binocular disparity are greater when, I'm sorry, that is incorrect. So ignore that last bullet. Convergence is greater when objects are close. Disparity is greater when objects are far away. All right, so some people have used our understanding of binocular disparity to create these magic eye images, which are images that if you can train your eyes to look at them in a way that infers depth, this 3D object will pop out at you while you look at this image. So this one is of the planet Saturn. So in the middle of this, there's a planet with rings around it. If you don't believe me, these were really popular when I was a kid, so in the early 90s. I spent a lot of time looking at these, so I can do it really easily now. But it does kind of take some practice to be able to see them. So if you don't believe me, just Google magic eye images, and there's tutorials that you can go through that will help you kind of learn how to see these, and maybe you can see the planet pop out. All right. So light's entering the eye. There's all this mechanics of the eye of just moving stuff around that we get a bunch of depth information from. But then that light needs to hit the retina. So light enters the eye through the pupil, goes through that lens to get focused so that it hits the back part of the retina in a focused way. And what's interesting about the retina, so this is a blow up of this little sliver here, is that the frontmost part of the retina is not where the receptors for light are. The light has to filter through all these layers of cells to get to this pigment layer of the retina in order to be absorbed. So the retina is kind of built inside out, in that the part that's furthest away from the front of the eye is where light starts the process of vision. So once light gets here, these photopigments absorb the photons of light, and that causes them to change their shape. When they change their shape, they start to open ion channels on the rods and cones that are the photoreceptor cells above them. So those are the receptor cells for vision. They're named after their shape, so the rods have this pointy bottom, the cones have the pointy bottom, the rods have square bottoms. But when their photopigments start to change shape, their ion channels were open, causing them to start firing action potentials. They send those action potentials up to these bipolar cells, which communicate to these yellow retinal ganglion cells. And these are the cells with the big axons that go into the brain through the back of the eye socket and create the optic nerve. We also have two other cells that allow for what's called horizontal communication, or the ability of information to be shared across the retina. So these horizontal cells connect the rods and cones together so they can communicate with each other. And these amacrine cells connect the retinal ganglion cells together so they can communicate with each other. So we have information going straight up and into the brain, but we also have information going side to side across these cells. There's kind of two interesting spots on the retina that we're going to talk about next. The first is what we call the blind spot. So you can see where the optic nerve leaves the eye socket. It's nothing but axons right here. So there's no photoreceptors to respond to light. So when light hits this part of your retina, you actually don't become conscious of it because nothing responds to it. And then there's this little divot in the retina here where the retina is less thick than it is in other places, and that's what we call the fovea. And the fovea is very important for our detailed visual processing. So let's talk about those two things. So again, the blind spot is where there's no receptors where information exits the eye. So where the optic nerve goes out into the brain, there's no photoreceptors there to respond to light. Now we're usually not consciously aware of these blind spots in each eye because of the offset of the images of the retina. So our brain does what's called completion. It takes the information from the left retina to fill in the blind spot on the right retina and vice versa. Right, so the first example of how the brain is recreating our perception of the world using a best guess, right? It's assuming that whatever's on the left retina probably matches what's missing on the right retina. Right, so that's why we don't consciously perceive that blind spot. All right, again, the fovea is this little part of the retina that's less thick. It's right at the center, so it's immediately behind the pupil. And it's in charge of what we call high acuity vision, which is just a way of saying high detailed vision. So whenever you want to process something clearly, you look directly at it, because that allows the light that it's reflecting to fall onto your fovea. There's also reduced light distortion at the fovea. Because it's less thick, the light doesn't have to filter through so many layers of cells, so it doesn't get as distorted as it does on other parts of the retina. Alright, so that high acuity vision is coming from the types of cells that we find at the fovea. So those two types of photoreceptors, are cones and rods, do different types of vision. So cones are good for high acuity vision in good lighting. So they do our day vision. And rods have really high sensitivity, so they allow for low acuity vision in dim light, or they are able to do our night vision. Okay, so during the day, as we're looking around and we're getting colors and textures and detail from the stuff we're seeing, we're pretty much using our cones to do that. And at night, or if we're looking out of our peripheral vision and things are kind of fuzzy and blurry and color information isn't good, we're using our rods for that kind of vision. The difference in their types of vision come from how they are connected. So cones have what's called low convergence wiring, or one-to-one wiring, which means that every single cone has a bipolar cell that it synapses with, and that bipolar cell synapses with a single retinal ganglion cell. So this is like high definition vision, where there's this one-to-one relationship between every photon of light that they pick up and a signal that gets sent into the brain. Rods, on the other hand, have what's called high convergence wiring, or many-to-one wiring. So you can see this whole group of rods synapse onto a smaller group of bipolar cells, and they all synapse onto a single retinal ganglion cell, which means that any individual photon of light that's picked up across this group is going to get smeared into this entire space, right? So we get low definition visions. The nice thing is that it doesn't take many photons to stimulate these rods, so they're highly reactive to light. They just always send kind of this blurry smeared signal, where cones need a lot more photons to get them stimulated, but once they start acting, you get this really clear signaling of what they're responding to. Alright, so from here we start to get into some important features for color vision that are happening at the level of the retina. So there's two theories of color perception. One is called the trichromatic theory, or also the component theory. So Young and Helmholtz proposed this theory, and they proposed that there's three types of receptors in the retina, each with a different spectral sensitivity, meaning that each are maximally responsive to different parts of the visible light spectrum. And they proposed that we had basically blue, green, and red receptors in the retina. And to support their theory, they basically demonstrated that by mixing combinations of those three colors of light, you could create pretty much any color we experience. Okay, so I know if you're thinking about paint, it's blue, yellow, and red that are primary colors. When we're talking about light, green is actually the primary color, not yellow. So if you combine equal amounts of yellow and green light, of red and green light, that actually produces yellow light on a stage. Right, so next time you're at a concert, look at the filters they have on the lighting for the stage, and you'll see that there's green and red filters that are creating the bright yellow colors that light up the performers. Alright, so the fact that we can perceive all of these colors and all of the variations between them does support that we probably have this kind of wiring at the retina. And it turns out that that is true, that we basically have three types of cones, they each have their own unique photopigments that are connected to them, and some photopigments respond to different wavelengths differently. So some respond more to blue, some more to green, and some more to red. Alright. The other theory of color vision is called the opponent process theory, which proposes that there's two different classes of cells for encoding color and encoding brightness. And each encodes two complementary color perceptions. So there's red-green cells, blue-yellow cells, and black-white cells. Okay, so to demonstrate this processing of color, I want you to stare at this image by focusing your eyes on the black dot in the center. Okay, and I need you to just hold your gaze there for about 30 seconds. So this is going to be awkward, but we're just going to wait 30 seconds. Don't move your eyes. It's okay to blink, but just keep your eyes focused on that dot. Keep focusing. A little bit longer. A little bit longer. Keep focusing, and I'm going to switch it to a white background. I want you to keep your eyes right there on the dot, and once you see the white background appear, just blink a couple times. What you should be perceiving is the American flag in red, white, and blue as you stare at this plain white background. Okay, so this is called an after image. It's an illusion. But basically it supports this opponent processing theory of color vision because it demonstrates that when we exhaust our photoreceptors for yellow, black, and green, and then we expose people to pure white light, which should stimulate all of them, they reflexively trigger the perception of their opponent color as they recover. So the cells that responded to yellow now make you perceive blue, the cells that were responding to the black now make you see white, and the cells that were responding to green now make you see red. So with the two theories of color, basically both have been supported and are correct. We have found that coding of color by the cones seems to operate on a purely component basis. So the three types of cones at the retina supports that theory. But then we see opponent processing of color at all levels from there. All right, so the retinal ganglion cells to the neurons in your occipital lobe seem to work on this opponent processing basis. All right. So once we have light hitting these retinas, it takes a pathway in the brain that we call the optic tract. So everything from our right visual field, so all the red stuff, projects onto the left side of the retinas of both eyes. And it stays on the left to get processed by the left occipital lobe. So the left side of the retina for your right eye crosses over to the left side of the brain at this point called the optic chiasm. Right, so just like our left motor cortex controls our right arm, our left occipital lobe processes visual input from the right side of space. From the optic chiasm, information goes to the lateral geniculate nucleus of the thalamus, so just a part of the thalamus. And from there, it goes to primary visual cortex, which is the back surface of your occipital lobe. Now, 90% of your vision goes this way. and that's how we get conscious processing of your visual input. You can see there's this tiny bit that's going off to the superior colliculus. That is 10% of our visual information, and that results in subconscious visual processing. So people who have damage to their occipital lobe have actually shown that they can still like duck if a ball's thrown at their head, because the superior colliculus basically helps you reflexively move in response to visual stimuli. It's a survival mechanism, right? So even though they can't tell you why they ducked or what was coming at their head, they still will reflexively duck without the conscious awareness of that information. Once we get to the occipital lobe, we'll find that there's several uniquely specialized areas for visual information. So there is a specialized area for motion perception, and we also can have motion after effects, just like we had with the after images we saw in the processing of color. So we've learned that this part of the brain also has opponent processing, where there's different neurons for up, down, left, right, forward, backward, and every variation of direction you can think of in there. So that's another principle of the brain, where you get neurons that respond to opposites, so that when they're excited, you perceive one version, say upward going motion, and when they're inhibited, you perceive the opposite of that, which in that example would be downward going motion. So if you'd like to experience a motion after effect, you can watch this YouTube video, and you'll see this illusory motion that it creates. I will warn you that if you're prone to migraines or visual headaches, I wouldn't watch it. It's a little bit trippy. And it does take a couple minutes for the effect to go away, so if you're not sitting still for a little bit, maybe don't do it, because it'll make you a little dizzy if you try to get up and move. Alright, so this is the entire visual system. Alright, so we talked about light going into the retina, coming down the fiber tract, the optic tract, to the lateral geniculate nucleus of the thalamus, projecting back to primary visual cortex, which is area V1 here. You can see there's all these other areas of visual cortex. That area for motion is marked as V3A here, and as V5 here, this little red dot. We also have specialized areas for color, such as area V4, right? So there's all this different types of information that gets extracted from visual input. And we know that it gets processed in all these different ways and eventually gets sent up to the parietal lobe. But again, the full binding problem we don't understand. We don't understand how all that information gets recombined to create our true perceptual experience. Just another note on this slide, extrastriate and striate cortex are other ways of referring to the occipital lobe. It just so happens that if you take a slice of tissue from the occipital lobe and stain it, the neurons are stacked in these layers that make it look like it's striped. So extrastriate just means striped cortex. It's another way people refer to the occipital lobe, so sorry if that's confusing there. Alright, so I started with that example of artificial vision from 2002. So we're going to end our discussion of vision by looking at this article. So I'm going to ask you to pull it up on your laptops, but what you should see is that basically we've developed these computer chip implants that can be put into people's occipital lobes, and they're run on like a little battery pack that they wear on their back, and they are responding to photoreceptors that have been implanted in these people's eyes. Right, so they don't have to wear any crazy goggles. Their eyes look normal and intact. And it's just, instead of actual photoreceptors in their retina, there's mechanical photocells. And they're responding to light, sending impulses into the occipital lobe. And because we understand the organization of the occipital lobe so well, we can recreate perception of the world that way. Now, these people can't read a book. right? They can't do high detailed visual processing, but they can walk around their environment without a cane, without a dog to help them navigate space. So they can like see enough contrast to see stairs and door frames, um, and the outlines of people. They might still have difficulty recognizing people if they don't hear their voice or something like that. But that's come a big way, where we're basically restoring people's independence by allowing them to see enough to get around and manage themselves without all of these assistive aids. All right, guys, have a great week. I will see you next Wednesday, and we will start talking about the rest of our senses as well as how attention influences our perception of these senses. I will see you then.